this is Dr. Launen, and I'm going to be talking to you about Illumina sequencing, a DNA sequencing technology that's also called Solexa sequencing and Illumina dye sequencing. It's a next-generation technology referred to by some as third-generation sequencing, with the first being Sanger and the second techniques like pyrosequencing, and it is the dominant form of sequencing found across the world today. It's used and useful for the sequencing of very large amounts of DNA, such as the DNA recovered from whole genomes and metagenomes. We're going to approach this topic by first, in this video segment, talking about sequencing by synthesis, which is the underlying sort of chemistry that drives the Illumina methodology, and it relies on what are called reversible dye terminators, a certain form of nucleotide that works in this sequencing platform. Then we're going to talk about library preparation and what the fragments of DNA within a library mean and what they look like. And we're going to then move on to talking about how to make clonal clusters and then look at the steps in sequencing the clonal clusters. And then you're going to need to take a nap or do something really fun because your brain's going to be hurting after you learn all about this really cool technology. All right, so sequencing by synthesis is something that the Illumina company owns or has patented, basically, and it relies on these nucleotides that are called reversible dye terminators. So here's a nucleotide, right? That base would be adenine, cytosine, guanine, etc., or thymine. Here's the 5-carbon sugar that we always see in DNA, deoxyribose, and here are 3 phosphate groups. So here's carbon 1, 2, 3, 4, and 5. Now in a normal nucleotide, you'd have an hydroxyl group here. And here instead of an OH, you see an OR, and that R is signifying the presence of what's called a reversible blocking group. So this is blocking the addition of another nucleotide temporarily, and that can be changed. So using chemical manipulations, once this nucleotide is added to a new and growing strand, you can stop the addition of another nucleotide by having the reversible blocking group there, and then at the moment that you want to change that scenario, you can regenerate an OH, which allows you to add another nucleotide to this position. So another feature of these reversible dye terminators are the dyes. And so the dyes are fluorescent molecules, and they are attached to the rest of the nucleotide with what's called a cleavable linker, which tells us that they can be removed and washed off, and that's done at each step of the sequencing in Illumina sequencing. And there's a different dye for each nucleotide so that flashes of color can be detected after we hit this structure with a laser beam, and the color will tell us what the identity of the nucleotide was. So these are really important structures in understanding this overall system. So here we've got the idea of sequencing by synthesis. So here is the template DNA running up here, here, and here. And here are 4 different types of reversible dye terminators, A, C, G, and T. And each of them has its little fluorescent dye attached. And so depending on the identity of the template DNA, the correct nucleotide will base pair in, and then you can hit it with a laser and it will fluoresce. Okay, and then you wash off the fluorescent dye and you regenerate the OH group, and then you repeat and add the next nucleotide to that position. And so the reversible part, like the blocking group, controls the timing of this, and the fluorescent part controls the detection. It gives us the identity of the nucleotide that's being added. Now, this particular image could be a little bit misleading because it's suggesting that very, very close together you would have 3 different templates, because you can see here 3 different nucleotides being added. But in fact, in Illumina technology, you will have little clumps of this called clonal clusters, and then at a different spot on a physical location called a chip, you would have a little cluster of this called a clonal cluster and a little cluster of that, so that you can image the total layout and see little clumps that are telling you that an adenine was added here, and then nearby there'd be another little clump that was lighting up with the guanine color, and close to that another little clump that was lighting with the C color. Each of them would be added at exactly the same time, though, in a cycle of sequencing that's controlled temporally by the blocking group and the timing of the lights and the additions of nucleotides. So sequencing by synthesis, if we look at it, here's another diagram. So we've got a template here, and in this scenario, we're sequencing in that direction. Okay, so here's our template. Here are our reversible dye terminators, and we've got the correct, whatever the correct nucleotide is, added on, complementary to this template. We hit it with light, we detect, and then what will happen is we'll wash off the fluorophore, the fluorescent dye, and we'll unblock the hydroxyl group on the 3' carbon. We'll add the next appropriate nucleotide, repeat by hitting it with light, detect it, wash the fluorescent bit off, undo the blocking and regenerate the OH group, then the next nucleotide can be added, and so on and so on. So we've got DNA polymerase that is present, and it is forming the phosphodiester linkages between each of the nucleotides in this growing strand. We've got laser energy, which is causing the emission of fluorescent light, and that's detected and used to identify the nucleotide. And we've got the blocker group chemistry at work, so that we're continually regenerating the 3' hydroxyl that is in a nucleotide added at the very end of the growing strand. And you just keep repeating this, and every time you do it once, it's considered one cycle. And in that way, you can use time to control how many nucleotides you sequence. So when we look at this in with the eyes of the Illumina detector system, we're detecting color, right? So you're looking at a chip. If this is actually a tile on a chip, and each of these little spots is one little cluster of identical DNA molecules that are working as a template. And so here, the authors of this figure are showing us this one spot here, 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 and here. And this is the same spot, but 9 different time frames, 9 different cycles of sequencing. And we can see that the first nucleotide was added, and it's giving a green light, and this is T. The next one that's been added is giving a blue light, and that's G. Then a moment later, we see a flash of red light, signifying C. We're back to a flash of green light, signifying T, and so on. Meanwhile, the same synthesis is happening in the clonal clusters at all these other little dots. And we can see that on this bottom dot that's also been circled, we've got kind of a dull sequence. It's not so interesting. We've got a string of T's, and then we've got a guanine, and then another T. And so this T and this T are added at exactly the same time in parallel, okay? This G and this T are added at the same time. In other words, they're added in parallel. And when you do that across literally millions of these little clonal clusters, that's a really big job to keep track of. And so it's referred to as being massively parallel sequencing, meaning that we're sequencing literally millions of fragments, one step at a time, and the timing is identically linked. So that's the idea of sequencing by synthesis, and it relies on an understanding of the reversible dye terminators and the idea of fluorescence as a detection system. So where is this going to fit into the larger process? Well, this is the sort of a little bit of skinny on the larger process for Illumina sequencing. You get your DNA of interest, maybe you extract it from microbes or another type of sample. You cut it up somehow into little chunks, and you make a library, and we'll talk more about that in a while, but basically that means a pool of small fragments of your DNA, hopefully containing all of your DNA in multi-copy, and on the outsides of those little fragments of DNA, we've got artificial sequences we're going to call adapters, and we'll talk about what they are in a future video in this series. Then we're going to take those little tiny pieces and we're going to attach them to a solid surface called a chip. And we were just looking at images of sequencing happening on a chip a moment ago as we looked at this slide. Then we're going to amplify each of those little bits of DNA into identical clonal groups. Clonal means identical, so little clusters of all the same molecule. And then we're going to actually do what is called sequencing by synthesis and detect the nucleotide sequences of our strands of interest. And then we're going to link the flashes of light with the position on the slide so that we know what the sequence of each cluster's DNA is. So we're going to know the XY coordinates of each of these strands, and we're going to tie that into the information that we get from sequencing, and we're going to use that overall in the process called assembly when we put all these little pieces of DNA together at the end to get back to what is our original target genome. All right, so we're going to move on in the next video lecture to talk about the rest of this now that we've talked about sequencing by synthesis.